Good day, televiewers, and welcome back to the program GC Revision Guide. Today we'll be taking a practical question as far as biology is concerned, that is 0510 and 0570. That is the code for biology. You can be asked this question. For example, what is photosynthesis? That is the part A, and the marks allocation is 5 marks. And at the same time, the mark allocation can also be 3. For B, you can equally be asked, how does photosynthesis occur in green leaves? 6 marks, or the marks allocation is 15. So today we are going to answer this question depending on the marks allocation. So when the marks allocation is 5, it means you have to give a maximum of 5 points to score 5 marks. And this is what you have to say. Photosynthesis is that process whereby green plants, green algae, and equally cyanobacteria manufacture their food is a point. Using simple raw materials such as carbon dioxide and water is a point. In the presence of sunlight which is captured by chlorophyll found in the chloroplast of the leaf is another point. During this process, a gas which is known as oxygen is given off as a byproduct. It's a 4th mark. And to score the 5th mark, you give a summarized equation of this process. Now, talking about the second part, which is B, if you are asked to explain how photosynthesis occurs in the leaf for 6 marks, this is what you are going to say. Your first statement, photosynthesis occurs in the chloroplast of the leaf. The raw materials are carbon dioxide and water. The carbon dioxide which is found in the atmosphere diffuses through the stomata to reach the leaf. The water which is found in the soil is being absorbed by the roots. It will pass through the xylem vessels to reach the leaf. And then light which is captured by chlorophyll will be used to split water into hydrogen ions and equally oxygen, which is usually given off as a byproduct. The hydrogen ions will combine with carbon dioxide and equally the endogenic acceptor of ribulose biphosphate to form glucose. This glucose is immediately converted to starch and it is stored temporarily at the level of the leaves. And in the night, this starch which is stored in the leaf is being broken down to a disaccharide which is called sucrose. The sucrose is carried in the phloem tissues down to the storage organs, which are the stems and the roots. When sucrose arrives these storage organs, the sucrose is converted back to starch, which is now stored at the level of the roots and the stems. So this in brief is how you can answer that question to score your 6 marks. Now, if the marks allocation is 15, it means that you have to go detailing. And you begin by saying that photosynthesis occurs in 2 phases. We have the light-dependent phase and equally the light-independent phase. During the light-dependent phase, there is hydrolysis of water in a process known as photolysis, and the light equally strikes chlorophyll. The chlorophyll becomes positively charged and electrons are released. These electrons, they are carried from one carrier to the other, and as they move from one carrier to the other, that is how energy in the form of ATP is being produced from ADP and inorganic phosphates. Equally, in a situation where we have 2 actional centers at the level of the leaf, that is where the chlorophyll molecules group themselves to the actional center, that is PS1 and PS2, light will equally strike chlorophyll at the same time. The electrons that will leave the first photosystem, that is PS1, which is at a higher energy level, will also move through electron carriers, and it will come back to the second reactional center, which is PS2, to stabilize the chlorophyll that was already positively charged. And in the second case, we'll have electrons which will leave water that was being hydrolyzed to stabilize the chlorophyll at the level of PS1. So in this case, we talk of a non-cyclic photophosphorylation because the electrons do not move in a cyclic manner. In another case where we have just one reactional center, that is PS1, when electrons leave chlorophyll molecule, the electrons are being carried from one carrier molecule to the other, and the same electron that left chlorophyll comes back to stabilize the chlorophyll as PS1, and this is known as cyclic photophosphorylation. So in short, in the first phase, which is the light-dependent phase, which takes place, of course, at the level of the thalocode membrane, there are two products which are formed. The first one is ATP, and the second one is reduced nicotinamide adenine dinucleotide phosphate. So that is the first phase, which is the light phase. Now, talking about the second phase, which is the light-independent phase, there are 4 sub-processes involved. And these processes are, you have fixation of carbon dioxide. We equally have the second, which is the reduction phase. We have the synthesis phase, and equally the regeneration phase. During the first phase, which is carboxylation phase, carbon dioxide, of course, which leaves the atmosphere, will combine with an endogenic acceptor of carbon dioxide called ribulose biphosphate to form a 6-carbon compound which is unstable and will immediately split to give 2 3-carbon compounds, which are 3-phosphoglyceric acids and 3-phosphoglyceraldehyde. So when we talk of C3 plant, it is because of these 2 products, because the first product of photosynthesis is a 3-carbon compound, that is glyceraldehyde, and equally 3-phosphoglyceric acid. That's why they call them C3 plants. Now, in the second phase, we have what we call reduction. And what is reduction? What are we reducing? The 3-phosphoglyceric acid has a lower energy value, so it is converted, it is reduced to 3-phosphoglyceraldehyde, which has a higher energy level. So that is the second phase, that is reduction of 3-phosphoglyceric acid to 3-phosphoglyceraldehyde. And the third phase, the third process, which actually involves the synthesis of glucose, is known as the synthetic phase. During the synthetic phase, 3-phosphoglyceraldehyde will combine with dehydroxyacetone to give you fructose 1,6-diphosphate. The fructose 1,6-diphosphate will transform itself to fructose 6-phosphate in the presence of the enzyme called fructose 1,6-diphosphorylase. The fructose 6-phosphate is isomerized to glucose 6-phosphate, and glucose 6-phosphate is equally transformed to glucose 1-phosphate with the help of the enzyme called glucose 6-phosphate mutase. And the last to give you your glucose is with the help of the enzyme called glucose 1-phosphatase, which removes the phosphate group from glucose at position 1. So that is how the synthesis phase occurs. And the last phase is the regeneration phase. The regeneration phase is very important because this process of photosynthesis has to continue. And what are we regenerating here? We are regenerating ribulose biphosphate. the endogenic acceptor of carbon dioxide. So it is through the 3-carbon compound, that is glyceraldehyde 3-phosphate and the hydroxyacetone 3-phosphate, that ribulose biphosphate is being established or being resynthesized after destruction. So in short, that is how we can go about answering this question to score a maximum of 15 marks. When we come back next time, we'll take another question, and maybe still on photosynthesis or on other related topic. Thank you for your very kind attention. See you some other time in this same program, GC Revision Guide.